All right, this is chapter 5, a chapter where we're going to focus on the structure and function of cell membranes. Cell membranes, often called plasma membranes, separate cells from their surroundings. But I want to make sure everyone understands that the plasma membrane that forms the cell membrane is also going to be describing the membranes that we find on the inside of cells. Some of the organelles that we saw, like the Golgi complex, vesicles, vacuoles, they're all composed of plasma membranes. Now, one of the things that's always amazed me is how thin these structures really are. They're only about 5 to 10 nanometers thick. And to put that into some common terms, it would take about 10,000 of them stacked one on top of the other to equal the thickness of a single sheet of notebook paper. So it's amazed me how thin they are, yet how structurally sound they are, and that they're able to withstand tremendous forces that our bodies put on our own cells. Think about the balls of your feet when you jump and land. Now, the main job of the plasma membrane that surrounds the outer boundary of cells is to control the passage of materials in or out of the cell. And a term called selective permeability is used to describe this in generalized fashion. In other words, a cell membrane being selectively permeable allows some substances to cross the membrane while others are not. Or some substances may cross easier than others. Now, the plasma membrane structure can be described by what is known as the fluid mosaic model. And prior to the 1970s, there were a lot of proposed models about how the cell's membranes or plasma membranes were actually put together. It was understood that phospholipids, those amphipathic molecules that form the phospholipid bilayers that we've discussed previously, were a huge component, as well as proteins. But what was unclear until the early 1970s was how these things were actually put together. And of course, some advances in microscopy and biochemistry have allowed scientists to understand those. So in 1972, two scientists, Singer and Nicholson, proposed this fluid mosaic model, where basically we have our phospholipid bilayer and we have proteins sort of forming this mosaic in the phospholipid bilayer. Some of the proteins are going to be passing completely through the phospholipid bilayer, while other proteins are attached to just one side. Now, the phospholipid bilayer allows for the membrane to be very fluid. What I mean by fluid is not necessarily a liquid, but think of it as being very able to absorb stress by motion, sort of like a wave, right? So the fluid mosaic model allows for the membranes to be fluid, and this helps to provide some of their structural integrity. Of course, the phospholipid bilayer itself is held together by the hydrophobic interactions of these fatty acid tails whenever we're in an aqueous environment. And these membrane proteins are allowed to drift or move laterally, but they very rarely flip-flop from side to side. So think of this as sort of being very motion-like, and it can move and absorb a lot of stress as the cell's membranes are undergo some of this stuff. Now, the phospholipid bilayer's fluidity is maintained simply because the fatty acid tails in most instances are unsaturated hydrocarbon tails. And you might recall from the discussions we had when we talked about saturated versus unsaturated fats, this is because of these double covalent bonds to the carbon atoms here. Now, these little kinks give this membrane some of its weakness, if you will, to make it fluid so that it can move. But in animal cells, we find cholesterol molecules embedded in this portion of our phospholipid bilayer. And these cholesterol molecules act to help reduce some of that fluidity to keep the cell membrane from just literally flying all apart, and also to act as a temperature buffer. Plant cells don't have that cholesterol in the plasma membranes, but of course remember that plant cells are supported by that external cell wall made of cellulose for their support.